I'm aware that so far on the auto-contouring review, most of the papers that we've looked at and the authors that we've discussed those papers with have come from the Netherlands. This week we go over the pond to talk to Xue Feng from the University of Virginia about his work on deep neural networks for thoracic organ at risk segmentation. And this paper comes out of his entry into the 2017 AAPM Grand Challenge on thoracic organ at risk segmentation. In this paper, Xue and his team propose a two-stage approach to performing that segmentation. The first stage is a localization network. This localization network downsamples the C2 image to a fixed volume size, around 200 pixels in plane and around 70 pixels craniocaudally, and uses a 3D UNet architecture to perform a multi-label segmentation to detect each of the organs at risk. The purpose of this first stage isn't to get an accurate segmentation, but it's to get a bounding box, a region of interest, around each of the organs at risk. Using that first stage, a region of interest is defined, and again the original CT image is resampled into fixed volume size inputs for a 3D UNet architecture. But in this case, the volume size is determined by the region of interest of that organ at risk, and different volume sizes are used for different organs at risk. Essentially, this second stage has 5 independent networks, one for each organ. And because a region of interest is being used, a higher resolution of CT image can be used for segmentation. So very much this second stage is around refinement of the segmentation and getting as accurate a segmentation as possible given the constraints of the graphics card. The output of these 5 different networks is then fused together and resampled to the original CT resolution to output the final segmentation. One of the things I liked about this paper is the fairly detailed analysis of different aspects of the network. I think one of the interesting things that they look at is the use of augmentation, both during training but also within their internal cross-validation. And by performing augmentation at validation time, they can further prevent the network from overfitting to the training set, which is quite important given that the training set for the AAPM Thoracic Challenge is fairly limited in size. They also report their performance using the test set of the AAPM challenge and using the metrics presented in that challenge. That challenge introduced a way of combining different metrics together to get a single score. We have a challenge with dice and, say, Hausdorff distance, that they have different meaning. So within the challenge, these measurements were merged together using measures of intra-observer variability, such that if a segmentation was perfect according to the ground truth, it would score 100. If it was around the same level of error as intra-observer variability would be around that sort of ground truth, it would score 50. And if it was twice as far away, it would score 0. And this two-stage network for the lung and the spinal cord scored well above 50. It's much closer, well, it's closer to the ground truth than general intra-observer variability. For the heart, it scored around 50, as did all of the other methods that were entered into the Grand Challenge. But the performance of the esophagus was quite poor, scoring only 27 in this normalized measure. Nevertheless, Xue and the team from the University of Virginia came second overall in that thoracic segmentation challenge at AAPM and have more recently submitted results that have taken them up to first place on that ongoing leaderboard. Let's look now at the interview that I had previously with Xue, where we explored some of the issues raised by this paper. Hi, Xue. Thanks for joining me today to talk about your paper looking at organ at risk segmentation using deep learning. To start off with, why don't you tell everyone a little bit about who you are, where you're at, and what your sort of current research interests are?
Sure, yeah. Hi, Mark. My name is Xue Feng, and I'm an assistant professor of research at the University of Virginia in the biomedical engineering department. And my research is actually both sides of medical image acquisition and also image analysis. And in acquisition, my research is focused on the MRI sequence development and reconstruction. And in the analysis part, I basically focus on the clinical applications and doing some image segmentation, registration based on both MRI and the CT.
So it's very much across the spectrum. So in this particular paper, you use a two-stage network, and I believe your motivation was very much from the limitations of the graphics card, that you have to do a two-stage network, one to do the local region of interest, so you can then do a finer resolution network around each organ. If memory was no object, do you think this is still an approach you would take, or is this a compromise?
Yeah, actually, that's a good question. And in fact, we developed this method about 3 years ago, and at the paper published over a year ago. And now that NVIDIA has much better GPUs with a larger memory and at a kind of affordable price, yeah, so I think certainly with a memory of say 24 gigabytes of affordable GPU, this motivation will be like less of the challenge. However, I think another motivation for the two-stage network in which we do the cropped image in the first stage is that we can also reduce the false positives by only focusing on the regions that contain the organ. Because otherwise, if you feed a very large image with a large field of view to the network, it might have like a false positives here and there. Well, there are some post-processing you can do, but it's more efficient to just eliminate the possibility of like segmenting the hearts in the on top of the lung like that.
Yeah, and then potentially, I guess you have the benefit of speed as well in terms of that second stage network. It doesn't have to process bits that are irrelevant.
Yes, that's correct.
And you noted in the discussion though that one benefit maybe of doing a whole network is taking into account that relationship between organs, and that once you crop it down for your fine scale network, that's no longer possible. All it's looking at is one organ. But the network seems capable of segmenting the organ anyway, because you're getting pretty good results. Do you think that additional context would help a network further? Or do you think actually, it's something that humans use, but a computer doesn't need?
I think that's a good question, and I think the answer is it depends on the application and depends on in the organ segmentation. Also depends on what organs are to be segmented. So yeah, let's take a look at what humans do segmentation. For example, in the thoracic region, when humans segmenting, say, the heart or the lungs, they don't necessarily need the location of a heart or the exact contours of the heart to segment the lung. Or when they are doing the esophagus and the spinal cord segmentation, the contours of the lung will not be a very important factor. Although the location will still be considered as some anatomical landmarks, it's not a key factor. However, if we look at some head and neck segmentation, some organs, they do not have enough contrast, and the segmentation of those organs, you might rely on all other anatomical biomarkers, and those sometimes align well with the contours of that external organ. So I guess my feeling is that for thoracic organs, it probably does not benefit to have the whole contours of other organs with respect to segment a specific organ. And in fact, we did do an experiment to compare these two because we cannot fit the whole resolution to segment all organs. We used the first stage network and compared running the network with multiple organ output and versus a specific organ output. And in the former case, we treated it's a multi-class segmentation, and in the latter case, we kind of merge all other organs together with the background, so that the information is still there, but we don't provide the exact contour information. And we found that the results are actually very close, and no statistical significance are found. So that means in thoracic region segmentation, adding other contours of other organs may not benefit the segmentation of this specific organ. And it does have other disadvantages of using organ-specific ones in this case, because you have to run a network for each organ.
Yeah, so it takes a little bit longer in that sense. Yeah. But I guess, as you say, for head and neck, there might be benefit because of that, it's harder to see the organs, and some structures, lymph node levels, etc., where they start and end are quite often defined relative to other stuff, as opposed to by an anatomical boundary. So very much depends on what you're trying to do. Yeah. I think one nice aspect of this particular study is you trained on the AAPM thoracic challenge data, but as well as testing on the test set provided by that AAPM challenge, you also tested on an independent data set from the University of Kentucky. When skim reading this paper, it confused me a lot because you kept talking about the UK data set, which in my mind becomes the United Kingdom. Yeah. But I thought that was nice, and it kind of highlighted a number of factors around clinical deployment that actually might not be revealed by just participating in a challenge, such as differences in contouring style. Do you think it's reasonable to expect a deep learning model to handle variations? I mean, particularly variations in contouring style, but also variations in imaging protocol, etc. How capable do we expect a network to be, or are we going to need to fine tune for each clinic?
This is a very good question, and this is indeed one kind of major challenge in using deep learning or AI in medical applications. And again, I don't think there is a single answer for all applications and all cases. I would still focus on the thoracic segmentation and like to talk about what exactly we found. So it was very interesting. So we just want to see how well it is applicable to a clinical data set that is not selected, but like we grab randomly from clinical practice. And yeah, we have collaborators at the University of Kentucky, and we collected about 30 cases there. And for all other organs except for heart, the segmentation performance is very good, and it is on par or even slightly better than tested on the challenge dataset. And then we take a deep look at why the heart had a very bad performance. And we did some analysis and found that it's very likely due to the fact of the respiratory control practice that's adopted in UK. Because in thoracic region, in the image acquisition, the respiratory motion is a main factor, and different clinics actually have different practices to handle that. And some use 4D CT, and some use breath holding, and at the UK they use belly compression. So with belly compression, what happens is all organs tend to cluster to the toward the superior direction. So that means the locations of a heart, it's is on average higher than what's on the challenge data set. And then we analyze a typical failure mode. And interestingly, the failure mode it mixed a lot of the liver region into heart. And if you think the way, because it's a compression, the liver kind of takes the location of the heart, and so it's the network just I think it just sort of remembers where the heart should be, and then it segments a lot of liver region. So yeah, so to address that, certainly we can try some augmentation or other techniques improving the network, but we found that those are not that efficient. And I guess as we all expected, the most efficient way is just to incorporate some local data, because those data sets, due to different acquisitions, are really hard to emulate by augmentation. And then we studied, okay, so how many cases do we need? And does it matter if we select the worst performing case, or some cases that they perform relatively good, or just on average, just random? And we found that by adding as few as 10 cases, this issue is largely resolved. And then we further compared whether using a transfer learning or training from scratch by kind of mixing these 10 cases with the challenge data set. And we found that these two options also doesn't matter that much. Either option would give a satisfactory result. But also it doesn't have to be chosen from the bad performing ones. And even we found that even we just add those that performed really good, it still helps to address this problem, because it's kind of force the network to look more of this type of images where the heart is on a more superior position. So yeah, and we had a follow-on paper to cover more details on this study we did. It was a very simple study, but we found that the results were very interesting and will potentially be helpful.
So it sounds like a paper that we should cover in a subsequent interview or video. I mean, really there, so because of the differences in acquisition protocol, it might be necessary to have different models for different institutions, or at least for different imaging protocols, and that there needs to be a certain amount of tailoring to the clinic implementing it.
Yes, that's certainly, yeah, I guess unfortunately that's what's maybe needed, but probably not every clinics, because yeah, I guess if we cover many different acquisition protocols and practices, the model will be robust enough. But still, because the acquisition technology is always evolving, so yeah, there will be new hardware, new practice. So yeah, I guess the model has to adapt with that too.
Great. Well, thanks for joining me today for this discussion. I'll look forward to reading the follow-up paper, which hopefully you'll be able to email me afterwards. So thanks very much, and I'll talk to you soon. Bye.
Thank you. Bye.
So for me, the most interesting question raised by this paper is really around whether the design of neural networks is a design compromise because of the limitations of the graphics card or the available training data, or whether we're making a pragmatic choice. The space of neural networks that we could explore, different designs, etc., is vast. And we need to make pragmatic choices to limit what we do so that we can get to a solution. As Xue highlighted in that discussion, he was influenced in his design by the limitations of the graphics card. But the design that he settled upon perhaps has some advantages in terms of the speed or the volume of training data he would require. So perhaps the pragmatic choice is also part of the compromise. Why don't you let me know what you think around this issue in the comments. And thank you very much for listening to or watching this week's video, and I hope you'll join me for the next auto contouring review.